In this video, we will be going over free response questions 3 and 4 from the 2015 AP Biology exam. The amino acid sequence of cytochrome C was determined for 5 different species of vertebrates. The table below shows the number of differences in the sequences between each pair of species. Question A. Using the data in the table, create a phylogenetic tree on the template provided to reflect the evolutionary relationships of the organisms. Provide reasoning for the placement on the tree of the species that is least related to the others. So in this video, I don't have the template here, just letting you know, but the diagram I'm going to be drawing is essentially the same as the template they provided. So we're going to start by finding the species that looks the least related, the most distantly related to the other species in this table. And it seems that that species is D. polylepis because it has 21 amino acid differences with E. ferus, 18 with G. gallus, 17 with A. forsteri, and 20 with E. africanus. So we're going to put it as the most basal group, the most basal branch on this tree. So now let's look at E. ferus. And if you can see in this table, it has 21 differences with D. polylepis, 11 with G. gallus, 13 with A. forsteri, but only has 1 with E. africanus. So this suggests that E. ferus and E. africanus are very closely related, likely in the same genus. And so we're going to put them as sister branches on our tree. And now that leaves us with G. gallus and A. forsteri. Now if you look at the table, you can see that G. gallus has 11 differences with E. ferus, it has 10 with E. africanus, it has 18 with D. polylepis, but it only has 3 with A. forsteri. And this suggests that G. gallus is closely related to A. forsteri, but since it has so many amino acid differences with the other species in the table, we can tell that it is distantly related to the species that are not A. forsteri. So this basically means that G. gallus and A. forsteri are going to be on a different branch of the tree from the other species, but they share a common ancestor. So if you can look on this diagram, G. gallus and A. forsteri are kind of two smaller branches off of one branch of this tree that's separate from the others. Question B. Identify whether morphological data or amino acid sequence data are more likely to accurately represent the true evolutionary relationships among the species, and provide reasoning for your answer. So one thing is that if you have two organisms that have a similar physical characteristics, that might not be because they're actually related. For example, birds and insects both have wings, but bird wings come from modified arms, forelimbs, and insect wings come straight out of their thorax, not from their limbs. So, and this is obviously because they evolve flight separately. Or you can have convergent evolution, such as the long-necked dinosaurs that had long necks to eat leaves on trees, and also giraffes have long necks to eat leaves on trees, and they evolved similar feature of having long necks because they were faced with similar selective pressures. Another thing is that amino acid sequences are almost always more similar in species that are closely related, because if the species share a more recent common ancestor, then their genomes wouldn't have diverged as much compared to species that are more distantly related. Especially with this protein in the question, cytochrome C, it's involved in many essential functions like cellular respiration and apoptosis. And so a protein like cytochrome C is probably highly conserved, and this means that drawing conclusions from comparing the sequences is fairly accurate, because this protein serves the same function no matter what the organism is. Moving on to question number 4. Part A. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell. Describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. So, when you have the interphase nucleus, the chromosomes are very loosely wound, but when a cell enters mitosis, the chromosomes condense into the familiar X shapes that you see in textbook diagrams, and that's because they're very easy for the cell to manipulate, and this makes it easier for the cell to separate the chromosomes evenly to make sure that each of the daughter cells has the appropriate number. In addition, meiosis 2 is essentially the same as mitosis, except the number of chromosomes is half of that of mitosis. And the chromosomes line up at the metaphase plate during mitosis and meiosis. And this is essentially to ensure that the chromosomes are evenly distributed as they are pulled apart during anaphase. And as I said before, the chromosomes are going to be pulled apart during anaphase by kinetochore microtubules. And the cytokinesis occurs after the nuclei have already reformed, which means that the nucleus is intact, all of the chromosomes are inside, nothing is going to get out of that nucleus, and it's safe for the cell to divide. Because if it divides before the nuclei have reformed, some things might slip through before the cells have fully separated. Part B. The genetic composition of daughter cells produced by mitosis differs from that of the daughter cells produced by meiosis. Describe 2 features of the cell division processes that lead to these differences. So in mitosis, you're going to have a diploid cell that has replicated DNA producing 2 diploid cells. In mitosis, you're going to have a diploid cell with replicated DNA, and that's enough to produce 4 haploid cells. And in meiosis, sister chromatids do not separate in the first round of division, because otherwise it's just going to be mitosis. If you have the chromosomes, if you have the sister chromatids separate, you're going to have twice as many chromosomes, which will leave the same number in each cell, the same diploid number. And finally, meiosis is sexual reproduction, which means that you're going to have crossing over between the chromosomes. And this is going to result in 4 gametes that may have different genetic makeup or the same genetic makeup than the 2 chromosomes in the original diploid cell, because they're going to be exchanging bits and pieces of the chromosomes. They're going to be exchanging the alleles. And so the 4 gametes that result will have different combinations of those alleles. Or they may have the same combination of those alleles as the parent cell did. Thanks for watching, and please like and subscribe for some more content.